All right, so we're starting chapter 11, DNA structure and function, and this is where I always tell students this is when the genetics class gets a little easier for you. Many of you have covered DNA structure and function for many years, maybe even since elementary school. So let's get started. It should be fun, right? All right, so one of the big questions about genetic material, oops, sorry, I'm trying yet another format here, so bear with me. One of the big questions in genetics was, what is the genetic material that passes information on from parent to offspring? And so in order to get at that question, we had to develop, or scientists had to develop some criteria. So here are the 4 criteria that were originally assumed that would be helpful in genetic transmission. First of all, you had to have a molecule that could carry information, enough information to make and to run an organism. So making, it's only one thing. Remember, organisms have a metabolism. They grow, they develop over their lifespans. So you had to not only make the organism, but maintain it. Transmission, the molecule had to be transmitted from parent to offspring. So it's something that could be passed on. The molecule had to be replicated or copied. So in other words, a faithful copy has to be produced from the parent to the offspring in order for the genetic material to be meaningful. And then the material has to be variable. There has to be variation so that you can have phenotypic variation within a population. That's a really big deal, right? So for many, many years, it wasn't clear which of the molecules inside of a cell was the molecule responsible for transmitting genetic information. So scientists assumed it could be either a nucleic acid, which, let me make my marker a little bit thinner here, could be DNA or RNA. They're both nucleic acids. Proteins, all right? Proteins are just long chains of amino acids. We'll get to that later. Carbohydrates, these are sugars. Okay, sugars in terms of large sugars like starch, or small sugars, things that taste sweet to us like glucose or sucrose. And then lipids, all right? Lipids are fats. So within all cells, we have lipids, carbohydrate, proteins, and nucleic acids. And I'm just going to highlight for a minute now. The top 3 are the only molecules that are used to make energy. That's supposed to be an R there. DNA and RNA, we don't actually get energy from consuming DNA and RNA, even though those molecules can be recycled in our cells. So that's kind of the first hint that nucleic acids have a different job. They're not used for energy metabolism. So in the 1900s, the very early part of the 1900s, scientists basically started doing experiments trying to understand which molecule is responsible for transmitting traits such as gender, hair color, body size, and what were chromosomes made out of? It turns out there's a whole lot of stuff in chromosomes, not just DNA. And so early on in the 1900s, we identified that DNA and protein are actually both components of chromosomes. All right, so big question was, what do we need within a system to demonstrate that proteins or DNA in chromosomes were the genetic material? Because like I said in the last slide, we figured out that DNA and protein were present in chromosomes. And we knew that chromosomes were transmitting information. So which was it? Protein or DNA that was going to be transmitting information? So a really smart guy named Frederick Griffith came up with an experiment. Several of them, actually. So the Griffith experiments are very important to understand. These are experiments that will, if you stay in biology, follow you for many, many years. You'll find Griffith's experiments in microbiology, basic biology, biotechnology, molecular biology, you name it. Griffith's experiments are almost always a part of the curriculum in biology courses. So Fred Griffith studied originally Streptococcus pneumoniae. It's a type of streptococcus, a little circular bacterium that you find very often in pneumonia patients. Now, bear in mind that pneumonia is not necessarily a disease. Pneumonia is a condition. It's a condition where you have fluid in the lungs. So this streptococcus gives you fluid in your lungs when it starts living in your lungs. It's a nasty thing, all right? Now, it turns out that there are two strains that Griffith was working with. One strain that he described as being rough, and the other one that is smooth. Okay, I'm going to stop here, and we're going to talk about what does rough and smooth mean, because if you don't know what that means, it's going to be confusing. At the time, we did have the microscopes that were good enough to actually see the surface of each individual microbe, but it was kind of difficult to do that. So what we were looking at, or what we look at when we are describing rough or smooth, is the phenotype of the colonies that these guys make on a petri dish. So this is my drawing, ha ha ha, you lucky guys, of a petri dish. Ooh, okay, didn't mean to do that. Let's see if I can zoom back out. I'll reset. Hold on. Okay, just reset. I'm just going to draw circles here. All right. Actually, that one's not big enough. All right. So going back to smooth and rough. Here are my representations of two petri dishes. Some colonies will look kind of rough. They have jagged borders, and that's what rough denotes, is the border phenotype of the colonies that these microbes build when living on a petri dish. The smooth strain made glossy, smooth colonies, okay? And so basically, in the rough strain, when you look at the individual cells, you'll see that they do not have what's called a polysaccharide capsule. It's kind of like a jelly coating on the outside of the cell. The smooth strain have a polysaccharide capsule. So what that means is that when you streak them on solid medium on a petri dish, they have a smooth phenotype on the outside. The rough don't. And so it turns out that in humans, the rough strain, our body, our immune system, our body's not protected. But our bodies aren't protected from the rough strain. The smooth, let me try describing that again. I'm jumping ahead of myself. With the rough strain, you have colonies that look rough, and because they do not have a polysaccharide capsule, the bacteria are easy to kill by our immune systems. The smooth have that jelly coating, and the jelly coating protects the bacteria from a human's immune system. So we say that the smooth are deadly. Okay, D-E-A-D-L-Y. On a whiteboard, it looks a little better, guys. On the other hand, the rough is going to make you sick, but you're not going to die from it, okay? So let's move on to how these experiments were done. Okay, so the experimental strategy. What we're going to do, or what Griffith did, was he took mice and he injected mice with what we're going to call type 3S, which are the smooth bacteria, or the type 2R bacteria. Now, remember the S, the smooth ones, were deadly. So when you inject a mouse, the mouse eventually dies of pneumonia. And when you look when you do an autopsy and you look at the mouse's blood, you find type 3S bacteria in high density in the mouse's blood. On the other hand, when Griffith injected type 2R, okay, the rough-looking bacteria, the rough phenotype of colonies, the mouse got sick, but the mouse did survive. There were no living bacteria found in the mouse's blood circulating. So, in that case, we call that a non-pathogenic strain. The S is pathogenic. When something's pathogenic, it's deadly, all right? Now, here's where the experiment got really interesting. Griffith took the type 3S bacteria and he did something called heat killing. In other words, he basically applied heat to the bacteria, killed the bacteria, right? Makes sense. It's why we boil water when you take water from a stream when you're camping. You try to kill the bacteria and any pathogens present, Giardia, cysts, things like that, so that you don't get sick. When you heat kill type 3S, the smooth, the pathogenic bacteria, the mice survive. Of Of course, because you killed it, right? And you don't find living bacteria in the mouse's blood. That kind of makes sense. Now, here's where things got really interesting. What Griffith did was he took some of this killed bacteria, okay, the type, oh, I did it again. Rats. Sorry. Rats, I guess I should say mice, right? So he took the type 2R, the non-pathogenic, the non-deadly, and mixed it with the heat-killed type 3S cells. And all of a sudden, the mice started dying. And when you look in the mouse's blood, the dead mice had type 3S bacteria. So what he did was he took the rough strain plus dead S bacteria, and you end up with deadly bacteria types, right? That is not a good thing. In fact, that explains a whole lot with mutation of bacteria and actually viruses, as you'll see later. So here's a graphic showing what happened, all right? You inject a mouse with the smooth bacteria, mouse dies. Inject a mouse with rough bacteria. Yeah, after several days, the mouse gets sick, catches a bad cold, but he survives. Now, the type S heat-killed bacteria, the type R heat-killed S were injected into mice. The mice survived. And then over here you have the type R, right? Live type R and heat-killed S. I wish they would have highlighted this a little differently. I'm going to put a little, okay, exit. I'm going to put a little red dot, hopefully here in these cells. Okay, I guess I'm not going to put a little red dot in here. Oh, I guess I can now. Little red dot in here. All right. The type S cells and the dead S and the living R resulted in the death of the mice. And then again, you isolate the type S bacteria from the dead mouse. And what this means is that something is going on to transform the non-pathogenic organisms into a pathogenic organism. So transformation, which was discussed in chapter 9, was the conclusion that Griffith came to explain why the type S was transforming into type 2R, transforming the type 2R into the type S. In other words, you took something that was not deadly and you made it deadly by adding this right here. And so it turns out that bacteria have the ability to absorb DNA from the environment, and that DNA can come from dead cellular debris. And so if you have living cells in the presence of dead cells, that can transform the living cells into something that the dead cells were able to do. Isn't that crazy? So it's really important when you're cleaning to wipe everything up. Don't just let it all sit there, okay? So that contributed to the idea that DNA was the genetic material passing on information regarding heredity. It was the genetic, because basically bacteria are real simple cells, super simple. They have not as many modifications as eukaryotic cells, right? Less lipids. They do have proteins and RNA and so forth, but there's less of it. So moving on, Avery McLeod McCarty in the 40s took that information and they decided to take it to the next step. So they decided to take our type 3S, bacterial cells, our pathogenic cells, and look at the actual macromolecules to make sure that it was a nucleic acid that was passing on information. Because again, you know, Griffith was pretty confident that it was DNA, but you still couldn't rule out RNA, all right? So they decided to fraction broken up bacterial cells and see if they could figure out which biomolecule was allowing the transformation event to happen. Was it a lipid? Was it a carbohydrate? Was it a protein? Or was it indeed nucleic acids? And could it be among the nucleic acids, either DNA or RNA? So what's kind of fun in the 40s, which we take for granted, to be quite honest, is that in the 40s, well, it wasn't new, but scientists were able to start using high-speed centrifugation. In other words, they spin things really fast to separate heavy items from light items. So if you have a test tube, let's say, I'm going to try drawing again. If you have a test tube and you get a bunch of cells in here and you lyse the cells, you bust them open, then you're going to end up with stuff just floating around in the solution, right? Well, if you spin the solution really, really, really fast, okay, here we go again. Sorry, folks. If you spin the solution really, really, really fast, all the heavy stuff sinks to the bottom, all right? And the light stuff stays floating. So that's exactly what they did. They made an extract that contained only DNA by spinning it real fast. And they were able to convert the type 2R microbes into type 3S, the rough, the non-pathogenic, into the pathogenic. However, DNA in that particular case could have contained RNA or some trace protein. So what did they do to account for that? This was brilliant, you guys. This is amazing. So it's easier to look at a graphic of the experimental design. So you have the rough cells, all right? You inject the cells, or you look at just the bacteria, and the rough cells don't basically transform because they're just rough cells. If you take type R cells and then you add a type S DNA extract, okay? In other words, you add just the DNA. The cells become transformed, all right? You inject it into a mouse, you collect the blood, and here you go, you get the cells. Over here, if you treat the DNA with an enzyme called DNase, which breaks up DNA, okay, makes it non-functional, you don't get any transformation event. The type R cells do not become pathogenic. Over here, if you use something called RNase, okay, it breaks up RNA instead of DNA, you use this solution in the presence of type R cells, you end up with cells that are transformed. In other words, you make the non-pathogenic pathogenic. Over here, protease is an enzyme that breaks up only proteins. So mix that with type R cells, again, you have a transformation event occurring. So what this experiment demonstrated was, if you get rid of RNA, so here you have no RNA. You know what? This pen is not as good as the last one that I was using. Over here, you have no protein, okay? No RNA, no protein, you have transformation. Only when you knock out DNA, only in the presence of DNase, do you knock out the transformation event. That's what gave us the real idea that it was DNA giving rise to the transformation event. In other words, it was DNA that was the transforming molecule. Now to even go farther, all right, that was just in bacteria. Maybe that was just a bacterial thing, okay? So Hershey and Chase decided, Alfred Hershey and Martha Chase, gave further evidence using something called a bacteriophage. A bacteriophage is a bacteriophage right here, T2 bacteriophage. We're going to be talking about that much more. This is a virus that only infects bacteria. It doesn't infect humans or other animals. It's a class of viruses that infect only bacteria. And these bacteriophage only have two molecules. Okay, now bear in mind, viruses are not alive. They don't change, they don't develop, they don't have a metabolism. They do not meet the criteria of being living things, okay? They have to live inside of a cell to do their job. They're basically parasites. They're molecular parasites. So bacteria viruses and human viruses and animal viruses are made of DNA and protein, only two components, all right? So let's see if we have a picture. Here's a bacteriophage. The proteins that are present are going to encapsulate the genetic material. In this case, it's going to be DNA. And we call this top part the head of the phage. This little central region here, this kind of barrel, is called a sheath. And then these little things that look like legs are called the tail fibers that help the bacteriophage stick to the surface of a bacterial cell. And then this base plate allows the virus to lodge onto the bacterial cell. So let's pretend this is a bacterial cell. If I can get my pointer to work here, my pen. Here's a bacterial cell, and these base plates stick to proteins that are present on the surface of the cell, okay, allowing DNA to be injected into the cell. So this is like Velcro. The base plate's like Velcro, allowing the bacteriophage to stick to a cell. So T2 and another phage called T4, we're going to talk a lot about this. We talked a lot about this in chapter 10. T2 is a virulent phage. It doesn't have a latent state. It immediately replicates when it injects its nucleic acid inside of a cell. It starts copying itself right away, and the cell bursts open, releasing new virus particles, new T2 phage. So this is what happens during an infection event, okay? The viral particle latches onto the cell. The cell itself has its own bacterial chromosome, right? The genetic material is injected into the cell. The genetic material is immediately expressed, and one of the first genes that is expressed is a gene that starts breaking down the host's chromosome. It just starts disintegrating it so that the cell can't do its own metabolic processes. Another gene that starts being expressed is one that starts making proteins to synthesize a new virus particle. The viral particles are put together, okay, they start being assembled, and then once the viral particles are assembled, the cell bursts, it lyses, releasing the new viral particles into the fluid surrounding the cell, and each one of these viral particles can attack a new cell. So you can start with one viral particle, right, and end up with thousands by the end of the day, killing thousands of new cells. So the hypothesis, the idea, is that only the genetic material of the phage is injected into the bacteria. How did they know that was the case? What they did was radioactive labeling. So it turns out that in DNA, and we'll talk about this more in another chapter, DNA has a lot of phosphate in it. So you can use radioactive phosphate to label new DNA that's being made, okay? Proteins don't have a lot of phosphate in them, so you know you're specifically labeling DNA. Sulfur, radioactive sulfur, becomes incorporated into proteins. So they used S35 to label any new proteins that were being made. They used the labeled phages to infect non-radioactive cells. You don't want to have radioactive cells there because then you have background and that's a mess, right? So what they did was, and this is really funny, you guys are going to laugh, they grew some cells in radioactive sulfur-containing medium and some with radioactive phosphate. So this was labeling proteins, this was labeling DNA, all right? Then they allowed T2 phage to be developed here. And what ended up happening was that the phages were going to either have radioactively labeled protein or radioactively labeled DNA. And then what they did was they allowed these radioactively labeled phages to infect different populations of bacteria. So what they were asking here is the protein injected inside of the bacterial cell, or is the DNA injected? Now, let's come back to this slide up here, okay? We already know that it's the DNA. So really what you're looking at here is radioactive proteins on the outside of the cell or radioactive DNA inside of the cell. We take for granted that we already know how this all works, but back in the day, they didn't know this. So what they were doing was they wanted to know, where was the radioactivity being maintained? Well, to do that, you have to shear the capsid off the surface of the cell after the infection event occurs, right? How do you do this? This is the funny part. They made bacterial smoothies. Gross, right? So they allowed enough time for infection to occur, and then they stuck either the S35 labeled phage bacteria cluster literally inside of a blender. The blender then sheared the phage capsid off of the bacterium. They did the same with the P32 labeled. Now remember, S35, sulfur, all right? That's protein. P32, that's DNA. They wanted to know which portion had radioactive cells, okay? Radioactive bacteria. Spun it down. The supernatant here, the liquid, contained the S35 and an empty phage. They were able to determine that the phage no longer had DNA in it. The pellet here, the heavier portion, contained basically DNA that had infected cells, but there was no radioactivity here. On the other hand, in the P32 labeled sample, you had clean lysate. That did have phage heads that had no DNA in it. These were unlabeled. All the radioactivity was inside of the E. coli cells here. So the assumption was that the injection of DNA into microbial cells, like what we saw here, carried the radioactivity inside of the cell. If the radioactivity was on the actual phage itself, on the protein portion, okay, that got sheared off. So let's come back here again. Let's look at this big picture again. If the radioactivity was on the purple part, it did not get put inside of the cell. Therefore, the cell was not infected. Therefore, that was not the genetic material. DNA, on the other hand, when you radioactively label it, that is what is injected, and that is what infects the cell and changes the cell's genetic traits. Delicious smoothies, right? You can measure this using a Geiger counter. This particular Geiger counter is not the type that you would use. So this is what the data look like, what the data bared out. So if you look over time, this was the agitation time, the amount of time they waited, 1 minute, 2 minutes before, minutes after infection, 3 minutes, 4 minutes, 5 minutes, 6 minutes. Look how fast the infection, or how quickly the infection happens. By the end of 2 minutes, pretty much you max out the total isotope in the supernatant. In other words, by 2 minutes, you have the maximum amount of infection. By 8 minutes, you have a little bit more, but basically you asymptote here. So most of the S35 was found in the supernatant. Most of the P32 was found inside of cells. There was a little tiny bit inside the supernatant, but the whole idea is that the P32, you found very little inside of the liquid portion. Okay, you found very little inside of the tube here, right here. And so what that means is that that was incorporated into the cells, and that was the information, that was DNA, not the protein, that was injected into the cells, and that's what remained inside of the cells. And so it was expected that DNA was probably the genetic material transmitting this new trait of pathogenesis into the cells. Now, things got a little more confusing in the 50s when people started looking at tobacco viruses. Now, why are we looking at tobacco viruses? Tobacco is a very important commodity. And one of the things that people noticed, excuse me, I'm yawning, that this particular virus, tobacco mosaic virus, a plant virus, was killing cells, but they figured out that it wasn't a DNA virus. In this particular case, what they did was they purified RNA in a similar process by getting rid of DNA and protein, and they found that this virus transmitted its traits through RNA. And so many viruses actually use RNA as a genome, not DNA. Isn't that a crazy thing? So we'll get back to that later. What we want to talk about now is nucleic acid structure. This is something that I will tell you right now will be on the next exam. You need to understand how an individual nucleotide is structured so that you can understand how DNA is put together. I know that a lot of you already have a good idea about, 5', 3', things like that. But, I have to admit, even through graduate school, I was still learning new things about the structure and function of DNA. It's an amazing molecule. Keep an open mind. Hopefully I'll hit something new for you and you'll find something, you'll take something new from this course. So nucleotides are repeating units. DNA and RNA are what we call polymers. They're made of repeating units of individual nucleotides. Nucleotides are linked to form a strand. Those links are going to be covalent bonds. We'll get to that later. 2 strands form DNA, and a single strand form RNA. So DNA is what we call a double helix. The double helix itself wraps and turns and makes folds. It bends, it interacts with proteins, making a 3D structure that we're familiar with, that double helix, right? So this is basically the levels that we're looking at. We have a primary structure, which is going to be the single strand of DNA. The single strand of DNA is made up of repeating units of adenine, guanine, cytosine, and thymine. These dots represent the ability to form hydrogen bonds. A's and T's can form 2 hydrogen bonds. C's and G's can form 3. When you have 2 strands of DNA, okay, here's strand 1, strand 2, sorry. They tend to form hydrogen bonds with A and T always facing each other and G and C always facing each other normally. There are other situations where you might have a G and an A. That's a mutation. We'll talk about that later. DNA itself then starts looping and packing. To take a molecule that, a single DNA molecule like chromosome 1 in a eukaryotic human cell, okay, is almost a meter long. You have to take 2 of those, coil them up tightly so that our cells can house them inside of a nucleus that is not visible by the human eye, okay? And we have 40 what of those? 46 chromosomes. Some are longer, some are shorter, but they all have to be packaged to be put away tightly inside of a very small space. And here's another thing about DNA. It also has to be packaged loose enough so that genes can be expressed. So DNA packaging is a really big deal in the world of gene regulation. So nucleotide structure, nucleotides themselves have 3 components: a phosphate group, a sugar group, pentose sugar group because it has a certain shape, and then a nitrogenous base. So this is what we're looking at. These are what the phosphate groups look like. You have a phosphate with a bunch of oxygens around it. 3 of those oxygens have a negative charge. These phosphate groups are what give DNA a negative charge. That's going to become important later. DNA as a molecule is negatively charged. There are 2 types of sugars. There's deoxyribose that you find in DNA, and then ribose. The difference between these 2 happens on what we call the 2' sugar, okay, the 2' carbon. Now, one of the things that is really important to understand about DNA is the magic that happens, DNA and RNA, right here on the sugar. So at each one of these points here, you have a carbon. With the exception of the top, you have an oxygen. So one of the most important drawings I learned as an undergraduate was this: oxygen, carbon, 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 and then another carbon up here for now. You have individual covalent bonds bonding the carbons to each other and to the oxygen, and then there's a covalent bond here between these 2 carbons. Organic chemists are really cool because one of the reasons they're really cool is because they have a tendency to label the carbons in clockwise order. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Let's see if I can make that look like a 5. All right. So you have the 1' carbon, the 2' carbon, the 3' carbon, the 4' carbon, and the 5' carbon. Sorry about that, guys. Zooming in, I didn't mean to do that. Okay, although it's okay because now we can look at it closely. Each one of these carbons have a different job. The 1' carbon is going to bond to what we're going to call a nitrogenous base. Above us, you can see guanine and cytosine. Each one of those is a base that contains a nitrogen group, hence a nitrogenous base. The bases are circular. They have 1 or 2 rings. We'll get to that in a minute. And you're always going to find the base on the 1' carbon on DNA or RNA. The 2' carbon is a real special spot here. You are either going to find on it a hydrogen. Let me see if I can make this an H. Or you're going to find a hydroxyl, an OH. OH is a hydroxyl functional group, okay? So if you remember your chemistry, this is one of the functional groups that is highly, highly reactive, all right? So hydroxyls on the 2' carbon is always going to give you an RNA, okay? An RNA nucleotide. That's supposed to say RNA. My pencil's not working great. Over here, the hydrogen is present on DNA. See if it gives me DNA, right? Boy, this pen isn't so good. I'm going to have to try to find the other one again, all right? So what we say is that DNA contains deoxyribonucleic acid, okay? You got rid of that oxygen. You deoxyified it, all right? That's the way I remembered it. So the 2' carbon in DNA always has a hydrogen. The 2' carbon in RNA always has a hydroxyl. That is a defining difference between DNA and RNA nucleotides. The 3' carbon is going to connect to the next nucleotide line. 4' is kind of uninteresting. It just holds everything together. And the 5' carbon is going to link to the phosphate group. All right. So I am going to turn this off, and we are going to go back to regular size. Wow, I really need to get a better pen. And that covers our differences between deoxyribose nucleic acid and ribose nucleic acids. Now, let's go to the bases. The bases occur as either single or double rings, okay? The double rings are called purines, okay? Now, it's kind of weird that that's a purine because I would think that a purine would be a single ring, but we're going to call those pyrimidines. Adenine and guanine are your purines. Thymine, cytosine, and uracil, which only occurs in RNA, are pyrimidines. And try to remember, what carbon do the bases attach to? The 1' carbon, all right? All righty, let's move on. So here is a nicer picture. I just always like to highlight that difference between deoxyribose and ribose. The reason why it's a big deal is that this hydrogen group right here confers a lot of strength, a lot of stability to DNA. DNA is a highly, highly, highly stable molecule. DNA is so stable that we can find DNA from a woolly mammoth that was frozen in Siberian ice, right? We've been able to, scientists, I always say we because, you know, I feel like I'm one of the scientists. Scientists have been able to sequence the mammoth genome by basically harvesting old DNA and from a frozen mammoth. They were around a long time ago, guys. But a lot of that stability is due to the double helix and the double helix design and this hydrogen here. RNA, on the other hand, is very unstable in the environment. If you were to go into a classroom and take a swipe off of the desk, you would find DNA, maybe not a lot of it, but DNA from students who had been sitting there before, from dead skin cells. Maybe they sneezed on the table, right? You would never find RNA. RNA is highly degradable, partly because it's single-stranded, but partly because of this very highly reactive 2' hydroxyl group. So, nucleosides are going to be the base plus the sugar, okay? Nucleosides are the adenine plus the ribose, which gives you adenosine. Adenine deoxyribose is deoxyadenosine. The nucleotide minus the base and the sugar is going to be adenosine monophosphate, AMP, adenosine diphosphate, ADP, or check it out, adenosine triphosphate, ATP. So basically, you get rid of different elements of the molecule and it has a different name. Adenosine alone is just the sugar with the base. Adenosine monophosphate, AMP, is a single phosphate, diphosphate, and then triphosphate. Doesn't that look familiar? Adenosine triphosphate is the unit of energies that our bodies use to, you know, do cellular functions, to move. This is energy, ATP energy. And ATP, I mean, there are a couple little modifications, but it's basically an RNA nucleotide, a modified RNA nucleotide. So that's one way that our bodies, you know, even though our bodies don't use ATP or don't use DNA and RNA to make ATP, it does recycle the adenosine to make ATP. All right, but it doesn't, I guess you wouldn't say you need the adenosine, but you need sugar and fat, carbohydrates and fats to fuel the production of ATP from ADP or AMP. That's a different class though, folks. So all nucleotides are linked together through what we call phosphodiester bonds. What that means is that the phosphate is going to connect the 5' carbon to the 3' carbon of another molecule. So you end up with what we call 5', 3' directionality. In other words, 5' is always going to contain, and I'm going to draw on this one here. The top of a DNA molecule does have a cap on it. We'll get to that later. But if you have a raw DNA molecule, you are going to have 3 phosphates on top. Okay, that's supposed to be a phosphate, and that's supposed to be a phosphate. This is a crummy pen. Okay, so 5' has phosphates hanging off the top. 3' has a hydroxyl hanging off the bottom. That's how you know the direction, top and bottom, 5' to 3'. And remember, the other strand runs in the opposite direction. It's upside down. So this is going to be the 5', and this is going to be the 3'. Okay? So you have what we call anti-parallel structure inside of the strand of DNA. 5' to 3' on side 1, top to bottom, and then flip it upside down on the other side, 5' to 3'. Okay? So you have anti-parallel structure here. So the discovery of the structure of DNA took a whole lot of time, and it took a lot more people than just Watson and Crick to discover the structure of DNA. Watson and Crick were very, very smart because they put together a lot of other people's experiments to discover the structure of DNA and how it worked. So the framework and the discovery had to do with Linus Pauling. Linus Pauling himself was a chemist who studied, in addition to DNA and RNA, or just molecules, he discovered the function of vitamin C. So you can give Linus Pauling a lot of credit for studying vitamin C. He took a lot of vitamin C over his lifetime, and he figured out that vitamin C was protective for our immune system. It helped our immune system function optimally. Rosalind Franklin and Morris Wilson they provided evidence that DNA was basically an alpha helix by using what's called X-ray diffraction studies. X-ray diffraction is a field of analysis where you basically trap a molecule inside of a kind of a piece of plastic, and then you shine X-rays through it to look at the shadow that the molecule casts. And then Edwin Chargaff, he came up with A's bind to T's and G's bind to C's, which is a really important rule, right? So basically, this is what Chargaff came up with, was in E. coli. If you have 26% adenine bases, you're going to have about that in thymine. 25% guanine, you're going to have about 25% cytosine. So what he noticed was that if you look at all these different preparations, you always balance A's and T's with G's and C's. There is some variability, okay, 26 and basically 24, but that just has to do with experimental variability. James Watson and Francis Crick came on the scene, and basically based on a bunch of data, they came up with a ball and stick model that explained the structure and function of DNA, or at least the structure. They came up with the function a little later. So in 1953, they published an article that described DNA. And in 1962, with Morris Wilkins, they got the Nobel Prize. Now, Wilkins published his work on the X-ray crystallography of the DNA molecule with Rosalind Franklin. However, unfortunately, in the 60s, women didn't get as much credit in science, in the field of science, so she was not a co-recipient at the time. After her death, however, the prize was awarded to Rosalind Franklin's family, so that she is now listed as a Nobel Prize winner along with Wilkins and Watson and Crick. So in their model, they proposed that there were 2 strands of DNA held together through hydrogen bonds. The hydrogen bonds were between A and T, 2 hydrogen bonds between A's and T's, and 3 hydrogen bonds between C's and G's. The nucleotide bases were inside of the molecule. A's and T's, G's and C's were on the inside of the sugar phosphate skeleton. The bases stack like dinner plates, and the sugar phosphate backbone is on the outside, like I said. The 2 strands run anti-parallel. The 5' end on the left side is the top, and the 3' end is the bottom on the left. On the right, you flip it upside down. The right strand is upside down compared to the left. 5' is on the bottom and 3' is on the top relative to the left-hand strand. The two strands twist around to form a helix, and the helix is right-handed. It spirals away from you, turns in a clockwise position. There are 10 bases and 3.4 nucleotides per twist. Within DNA, we have what's called a major groove and a minor groove. This is the major groove, and this is the minor groove here. This allows enzymes and proteins to bond to the DNA molecule within the major groove. There's not a lot of bonding in the minor groove. We'll get to that later. Here's the top of strand number 1. This is the top of strand number 2. Remember, anti-parallel. And you have to remember that these are relative to one another. When we say relative, you're comparing one thing to another thing. You're comparing strand 1 to strand 2. So strand 1 has a top side. Strand 2 has a top side, but compared to each other, they run anti-parallel. A's, T's, G's, and C's always bond on the inside, holding strand 1 to strand 2. These hydrogen bonds are not the strongest bonds in the world, but when you have millions of them together, they hold the molecule together very, very strongly, making it a very stable molecule. So here's the minor groove when you look at a ball and stick model, and here's the major groove. And again, the major groove, it's called the major groove because it's wider than the minor groove. This is where you're going to find a lot of proteins and enzymes sticking to the DNA molecule, allowing the DNA molecule to be packaged and to express genes. We'll get to that later. The sugar phosphate backbone is highlighted here in green. This helps protect the base information inside. So here are the bases inside. Here's the sugar phosphate backbone on the outside. So as you can see here, there are a lot of different ways to look at DNA. You can look at what we call a ribbon model, so that you can highlight the sugar phosphate backbone separately from the bases. You can look at the ball and stick model. The ball and stick model, or the space filling model, give you an idea about the relative distances among the different molecular groups inside of the molecule. So it's taken all of these different representations to get a good handle on what does DNA look like and to express its functionality. So this is a field that's really important. X-ray crystallography, biochemistry, but also just the artistic representations are a big deal. The creativity of people's representations allow us to understand how the molecule works. So for the artists out there, big shout out for you guys. Without the artist representation, we wouldn't have as strong an understanding of what the structure and function of DNA are without being able to visualize it using our artistic tools and imagination. So there are different types of helices. All right, we've looked at DNA as an alpha helix, but there is also a B DNA and Z DNA. DNA is a double helix that can form what we call secondary structures. The predominant form that we find in living cells, even though we know the alpha helix is that simple form that we just looked at, bDNA is present in most living cells. zDNA and aDNA are a little bit different. In vitro conditions, in other words, test tube conditions, give rise to aDNA and zDNA. aDNA is similar to bDNA. It's a right-handed helix. However, it occurs in low humidity. Basically, what it is dehydrated DNA. It doesn't have a whole lot of biological importance. It's probably just an artifact of low humidity. zDNA is DNA that turns in the opposite direction. It's a left-handed term. It may have something to do with like recombination in yeast. You can find it in high salt or low salt concentrations, but basically you don't find this very often. You find zDNA in very unusual conditions. So when you're looking at bDNA, this is normally what we look at. Here's the space filling model. zDNA twists in the opposite way. So RNA does have a slightly different structure. It's copied from DNA during transcription. We're going to talk about that later on. RNA synthesis, which is the transcriptional process, only forms one strand from the double-stranded DNA. The primary structure of RNA is much like that of DNA. However, it's only one strand. And RNA typically has only several hundred to several thousand nucleotides in length. DNA can be millions of nucleotides in length. So RNA is much smaller. Okay. RNA also has a 5' to 3' orientation, but look, there's no other side here. 5' is always the top with the 5' carbon containing the phosphate. The 3' end is always the 3' carbon here with a hydroxyl hanging off the bottom. And remember, in RNA, we have that 2' hydroxyl, conferring a lot of instability to this molecule. They can form short double-stranded regions. We're going to talk about that. That's called RNA snapback or RNA base pairing. Basically, the idea is that RNA helices are going to be a right-handed helix, and that happens when RNA snaps back on itself and forms hydrogen bonding. We'll get to that a little bit later. So those are going to form secondary structural structures. Tertiary structures can happen when you have a really long strand of RNA forming these bulging loops, and the RNA strand, if you think about the RNA forming a bend up here, snapping back on itself. You can have internal loops when RNA snaps back, multi-branch junctions where you have strands of RNA coming together, or a single really long strand forming this type of structure, and then a stem loop. You find this very often when you have super long strands of RNA as well. Non-complementary regions are always going to bulge out. Complementary regions are going to form tight hydrogen bonds. So basically, these are stems and these are loops. Transfer or tRNAs have a lot of stems and loops. We're going to talk about that a little bit later, but that allows them to do what they need to do. The transfer RNAs interact with messenger RNA. So a transfer RNA, which, again, we're going to talk about in detail how it works, contains these ribbon regions, which are going to be hydrogen bonded regions with other sides of the same transfer RNA. And then you have these regions here that just kind of form these loops here. Down here, this is a double helical region. This is a double helical region. The 3' end right here, the acceptor site, is going to carry an amino acid. The 5' end just kind of hangs out there. And then this is the anticodon site that's going to interact with the ribosome. We'll get to that later. Okay, you don't need to know all the great details of that. This is where we're going to end this particular lecture. We'll get to more detail about the RNA in another lecture. Please be sure to go and do the homework and really, really, really focus on. This is one of the things I can't stress enough. The differences and the similarities between RNA and DNA. And a lot of it lies right here in the sugar. I know we spend a lot of time with ATs, GCs, and Us. That is a characteristic that is different between DNA and RNA. No T's in RNA, you only have the uracils. But I really want you guys to focus on these sugars, the deoxyribose and the ribose, because that's kind of a game changer when you're looking at functional groups and the functionality of the molecule.